My name is Kinaan Taha. I'm a chemical engineering student and a senior here at the University of Minnesota. And for the past summer and a little bit of the semester before then, obviously before COVID, I've been working with the Pomerantz lab studying protein-protein interactions specifically related to bromodomains and bromodomain binding. So bromodomains are a family of human proteins that have been linked to heart disease and cancer when left to their own devices, essentially. So a lot of the research in the Pomerantz lab has been focused on regulating and adjusting the binding of bromodomains by generating ligands to bind them and competitively inhibit their interactions with other substrates. My research specifically has focused on sort of two sections. One being a computational screen of 34 human bromodomains to identify their binding sites and which of them, if any, have more than one binding site, the one expected binding site for them to bind their own substrate. The other section of my research has been focused on the optimization of a synthesis reaction for a thiazepine scaffold, which is closely related to a previously discovered ligand for BRD4, the first bromodomain or first domain of BRD4, shown here in figure 1. So I essentially, for the synthesis, modified this structure by changing the functional groups and importantly introducing a double bond or alkene group into the 7-membered ring structure, changing its 3-dimensional structure, and hopefully leading to different binding characteristics between it and different bromodomains. Pictured here in figure 2 is sort of a typical and often studied bromodomain structure. This is BRD4, which has been previously studied, and like I said, is what this first substrate has been found to bind to. You can see how it has sort of these main 4 alpha helix structures forming kind of a column in the bromodomain, which will be interesting later. And then in figure 3, I've shown the planned synthesis omitting reaction conditions for the thiazepine scaffold that I worked on this summer. The first step being an esterification reaction to get it into this form with this trifluoro substituted carbon on the end here. And then cyclization, which is as far as I've gotten this summer. Hopefully next semester I'll be able to work on the reduction in order to get rid of that ketone group in the top right. So on to how I did my screening. I used a program from Boston University called FTMap. Anyone can use it who has a university email account. It's free online. And I basically just submitted all 34 of my bromodomains that were a part of my screen. And FTMap used computers at Boston University to generate a map of small probe molecules that were used to probe the binding sites of the bromodomains that I submitted. So it essentially threw a bunch of small solvent-like probe molecules at the protein that I submitted and found areas where the probe molecules sat and were able to bind to the site in a way that indicated possible binding hotspots. The threshold that I used for considering a hit was if there was a cluster of probe molecules that had at least 16 small solvent-like molecules in one spot. A kind of visual representation of this, because I know it kind of sounds a little bit abstract, is right here, where I've reproduced the same protein that I've shown over here, BRD4, and added the clusters that were discovered from the FTMap probe. So the 3 main clusters are highlighted and labeled. And this was essentially the form that all of my FTMap computational screening took. It was submitting the proteins, finding the clusters, identifying them, and then just counting up how many molecules were in each slot and taking that data down. My synthetic research, the methods included first following the previously discovered synthesis process that was used to create, to get this molecule, which is I think called ligand 1 in this paper. And then finding that those didn't really work for making the molecule with propiolic acid, so that had that alkene group in the 7-membered ring, and eventually leading to optimization of the synthesis reaction, including changing the coupling reagent and omitting one of the other reagents that was used for the esterification. And as of yet, I haven't been able to do the same optimization process for the cyclization reaction, so I'm still getting low yields on the cyclization, but I have been able to successfully generate cyclized product So that's this molecule down here in figure 3. The results of my study were that with the computational screen, out of 34 bromodomains, I found that 33 showed up as having hits from FTMap for at least one binding hotspot, which is expected because these bromodomains are essentially designed to, bind a substrate or one of several substrates in the human body. So the fact that almost all of them had at least one hit was a good sign, and it aligned with my hypothesis that they'd each have at least one. The only one that was a miss only had one probe molecule fewer than my threshold, so still encouraging data. The more interesting finding was that 26 out of 34 bromodomains had at least 2 binding sites. So there were a good majority of the bromodomains that I studied that had more than one binding site, which is really encouraging because it means that there are other avenues for selectivity when designing ligands for binding these bromodomains. So if we had a therapeutic treatment or a drug molecule in the future that could bind a bromodomain and hopefully be used as a therapy, it would be less than ideal if it also bound other proteins or other bromodomains and caused, other kinds of side effects or just not being as effective as it could be. Of those 26 with at least 2, there were 11 with at least 3, and I found 2 molecules that had 4 probe molecule clusters with more than 16 probe molecules. My findings for the synthetic optimization of the esterification reaction are in table 1. You can see that originally this first row is the unmodified synthesis process that was used for the esterification, leading to this ligand 1 up here in figure 1. And we had a yield of 3%. So obviously there was some work to be done to increase that. By first trying to reflux it, we did increase the yield up to 19%, but we're still kind of unsatisfied with that. And so after consulting with some other people in the lab and with the help of my laboratory mentor, Jennifer Kimbrough, we changed the coupling reagent to DCC, which made the yield go down until we realized that triethylamine was only being used as a reagent to stabilize the EDCI complex with to absorb the hydrochloric acid that was generated when the EDCI was used as a coupling reagent. And so by omitting the triethylamine, we were able to get our yield to jump up to 51%, which was held over 3 different trials, I think 2 of them in duplicate, with different scales. So it worked from 100 to 250 with the same yield percentage. And then I unfortunately did this after the table was made, but 750 and a 500 scale, 500 milligram scale experiment that had a similar yield. So moving on from the esterification, once we had a good way to make the ester, we tried the same conditions in the paper that I cited here in the bottom for the first citation, also from the Pomerantz lab, to cyclize that esterification product. Also, unfortunately, completed after the poster was submitted, but I got a 14% yield for the cyclization product, which was encouraging and obviously merits further optimization in order to increase the yield, but also further experimentation to discover whether or not this thiazepine scaffold is actually useful for binding bromodomains, and if so, which ones it is selected for, if any. So hopefully to continue this research, I'll be able to capitalize on the computational screen that I did by finding out whether or not these other binding sites are useful for selectivity analysis, for generating new ligands to fit into those binding slots, whether or not the binding sites are even real or just artifacts from the FTMap screen. There's a lot to be done in order to take this computational screen into the next steps of research. Obviously, right now with COVID going on, the computational screen has been good to be done remotely, and I've been able to find some pretty cool stuff from just the work that I've been able to do online. So hopefully next semester with UROP, the UROP grant that I've gotten, I'll be able to continue my synthetic research to generate other cyclized thiazepine products related to this scaffold, as well as hypothetically, hopefully, being able to test their affinity for different bromodomains, including BRD4. Thank you. If there are any questions, feel free.
Thanks, Kinaan. That's really nice work. If I remember right, bromodomain proteins are thought to interact with chromatin and affect transcription.
I believe so, though I've been focusing more on the specific binding with artificial ligands than the role of the bromodomains in the human body naturally. But I believe you're right. It's something about chromatin and lysine residues.
Okay. Is the natural ligand known?
Uh, there are 61 human bromodomains. I do not know how many of the natural ligands are known and identified. A lot of the crystal structures that I used for my computational screen were from bromodomains in complex with either other proteins or other ligands. So
Okay.
some of those may have been the natural substrate, and some of those may have been other designed ligands to bind those sites. I'm not 100% sure.
Because presumably your artificial ligands are based on some natural ligand that somebody found somewhere, right?
Um, I think that the current state of of discovering ligands for bromodomains is through fragment-based drug discovery. So more
Okay.
finding, yeah, sorry, I didn't mention that during my presentation, but more of the method of drug discovery has been finding a large library of small molecules and then
Yeah.
using SAR by NMR to eventually hopefully be able to link them together using linkers of some sort and design a drug-like molecule. I don't know how much of the space. I know that the Pomerantz lab has mostly been working on fragment-based drug discovery. So building a library of small molecules in order to build larger substrates.
Okay. Yeah, that sounds good. And then once you synthesize the substrates you're interested in, what binding assay will you do? I presume you'll check for binding to BRD4.
Yes, actually, there's an interesting paper that's not cited in this poster, but a lot of other research at the Pomerantz lab has been on protein-observed fluorine NMR. So taking bromodomains and selectively fluorinating certain residues, and then using protein-observed fluorine NMR to um, check for shifts in the chemical space surrounding those fluorine residues to determine binding. So hopefully in the fall semester, I'll be able to use either that or I believe one of the other methods that we use for determining binding has been, besides protein observed fluorine NMR assays, has been CPMG, competitive CPMG studies, checking the rotational properties of ligands when bound and when unbound.
Great, thank you.
Mm-hmm.
Kinaan, what is reflexing?
Refluxing is just when you heat a reaction that's undergoing and you have a cooled sort of condenser above it in order to ensure that you're not losing actually losing solvent or product molecule. So it's just a controlled way to increase the temperature of a reaction in order to push it in one direction or the other.
Okay.
That is a good point. There is not a data point for refluxing using the DCC conditions.
You didn't do that. You didn't include it, or it just wasn't done.
It wasn't done. I was pretty happy with 51% yield and wanted to move on to other aspects of the project.
Yeah. Yeah.
Thank you. Really well done. Such interesting work.
Thanks.